sounds like something that could be from science fiction.
These are immortalized cells.
Here we can see many different mini-tumors growing in this dish. This is a really robust case, and we know that it's resistant to a lot of therapies, so we're interested to find new treatments. I'm Jennifer Rosenbluth. I'm a breast medical oncologist. I treat breast cancer patients. I specialize in breast cancer, and I also run a lab that utilizes organoid cultures to model breast cancer and breast cancer risk. A lot of people think of organoids as mini organs, or in our case, mini tumors that are grown in a dish. And that is true in a sense, but they're really a simplified system.
They're alive and growing well. What's cool about these ones is that they're also fluorescent.
You can generate some organoids using a type of stem cell technology, or you can generate organoids from a tiny piece of a normal tissue or a tumor tissue.
You can see like the nucleus.
They're a way for us to take tumor material from a specific patient's tumor, grow it in a lab, make miniaturized versions that we can then use to do things like test drugs, model, study in real time now in a laboratory dish. As I see in the clinic every day, patients with tumors of the same subtype and same features can respond very differently to the same drug. And now we have a model system that really lets us study that in the laboratory. Once an organoid is established, we can replicate that tumor and make many, many mini-tumors, and now we have sufficient numbers to test even large number of drugs. In this study, we identified that most clinical biomarkers can actually be evaluated in organoids. So a biomarker that predicts response to a specific therapy also predicts response to that therapy in the organoid model. If you know there are 3 drugs that might work for that patient, now you can actually test those 3 drugs in a miniature tumor model from that patient. And maybe you can say, hey, of those 3, this is the one that has an amazing effect. You can see the tumor cells dying as you give them therapy, so it's a very visual experience.
You see already within 12 hours they start to fall apart, and that is because in this condition we added a drug that was very effective in killing these organoids.
Before this experiment, Tambin could add really high concentrations of chemotherapy and it wouldn't touch them. They'd just be perfectly happy. So here she's identified something that's finally able to kill these really resistant tumors. There's 2 different kinds of research. One is discovery research, so finding new therapies that are going to work for many patients. And the other is the idea of personalized medicine, where you're testing a drug to identify the drug for that specific patient from whom that organoid is derived.
This is an experimental drug, and it's like pretty potent against cancer. The reason why we don't use it in people is because the moment it gets in low temperatures, it clumps up into this crystal at the bottom. So I'm trying to shake it to the point where the crystal goes away.
UCSF is at the forefront for personalized breast cancer treatment through our large clinical trial programs, and now we're finally able to integrate organoids into that framework and hopefully improve our clinical predictions.